Dear student, I am Dr. 𝗓𝐴𝐻𝐵𝐞, Professor of Biochemistry. In this lecture of connective tissue, I will be going over collagen structure, collagen biosynthesis and applied clinical aspects. Let's get into the details of this lecture. You know, extracellular matrix, which is referred as ECM, it is also known as connective tissue. Now, the extracellular matrix, it contains three major classes of biomolecules which can be classified as structural proteins, specialized proteins and amorphous ground substance. These three things together makes an extracellular matrix. Now, among the structural proteins, we have collagen, elastin and fibrillin, and in a specialized proteins class we have fibronectin and laminin, and in an amorphous ground substance category proteoglycans comes in along with water, minerals and some of the small molecular weight proteins. So collectively all of them will make extracellular matrix, which is referred as a connective tissue. Now, one of the important component of that fibrous element or the structural proteins that are present in the extracellular matrix is collagen. Collagen is the major structural component of most connective tissues. Approximately 25% of mammalian protein in the body is collagen. And collagen is a fibrous protein and that will possess a high tensile strength and the collagen cannot be stretched. And there are at least 28 distinct types of collagen that are found, and all these 28 distinct types of collagen, they are made up of 30 types of polypeptide chains which are referred as alpha chains, and each one of them are coded by separate genes. Now, here is the table just for your information that how these 28 distinct types of collagens are made with specific genes that are coding for each one of them, and structural details of all these different types of collagen and their location is given. This is just for the information. It will be difficult to know everything or memorize everything from this particular table, but this is just for a reference, because as we move on in this lecture, I will be explaining you biosynthesis of collagen and some of the important types of collagen and their location and the clinical aspects. So otherwise, you know, type 1 collagen, just to go over a few of locations of few collagen molecules. So especially like type 1 collagen, it is found in skin, tendon, bone and cornea. So as a general mnemonic or general idea to remember type of location of type 1 collagen, if you look for one in all these tissues, like skin, can write it as skin, just to know that it is type 1 collagen, or tendon, write it as tendon, tendon, that is 1 there, like that. Bone has 1, so that is type 1 collagen. Cornea, so some like can make 1 there, cornea 1. So like that, roughly you can remember a type 1 collagen location, like skin, tendon, bone, cornea. And type 2 collagen, it is found in cartilaginous tissue and vitreous humor. So you can remember as cartilaginous tissue, that is there is 2 there. Instead of writing cartilaginous tissue, write it as cartilaginous tissue. So there is 2 there. And vitreous humor, instead of writing that vitreous humor, so vitreous humor, so there is 2 there. So like that, can make some rough idea how to remember the location of each type of collagen. At least first one 1 to 4 types of collagen. And number 3 collagen, type 3 collagen, it is found in skin, muscle associated with type 1 collagen, and usually it is found in reticular fibers. You can remember reticular fibers, there is 3 there. Type 4 collagen is generally found in basal lamina and the basement membrane of the kidney in the glomerulus, and also basement of stratified squamous epithelium. So we use the term basement there. Basement means under the floor, that is 4, right? Just a rough idea, under the floor, under the 4. So that's how you can roughly remember type 4 collagen is present in the basement membrane. So this is some of the example for some of the collagens which are located in certain organs and certain tissues. So moving on, looking at structure of collagen. So all collagen types have triple helical structure. That's first point that you should remember here in terms of structure of collagen. And approximately about 1000 amino acids are present in each alpha chain, and each alpha chain is twisted in a left-handed polyproline helix with 3 amino acids per turn. So the alpha chain direction is in the left-handed polyproline helix. Remember that polyproline, many prolines comes in the alpha helical chain, thereby there will be introduction of bends in the alpha chain, and there are 3 alpha chains that will wound around each other in a right-handed triple helix. So you can see here left-handed polyproline alpha chain, 3 of them they wound around each other in a right-handed triple helix, and because of this left-right combination, so left-handed alpha chains will be wounded around in a right-handed triple helix. That will make a triple helix collagen molecule difficult to unwind. Okay, that's where is the strength of collagen also comes from. So collagen alpha chains are difficult to unwind because of the opposite in directionality there. And also know that triple helix structure of a collagen, which is a rod-like molecule, and the diameter of the triple helix structure is 1.4 nanometer, and the length of each that collagen fibril is 300 nanometer. And amino acid glycine, which is the simplest amino acid of all 20 standard amino acid, it is present in every third position in a collagen molecule, and that is one of the unique feature of a collagen, because every third amino acid in a collagen molecule is glycine. And also collagen fibril is arranged in a quarter staggered alignment, because each collagen fibril, it will overlap at least 1/4 of its length with the other collagen molecule. Like that, there will be overlapping of each collagen fibril alpha chain fibril with other collagen fibril. So that's how a triple helix structure and collagen fibril is made. So that quarter staggered alignment is one of the characteristic feature of collagen molecule. Alright, now let's get into structure of collagen molecule. So as you see here, the amino acid sequence of collagen, it is written as glycine XY sequence, and this glycine XY, as you see, it is repeating. Glycine is appearing in every third position and then followed by X and Y. So what is there in X and Y position? So glycine X and Y repeats n number of time. So about 1000 amino acids are present in an alpha chain. This is at least 1000 amino acids in an alpha chain. In that, 1/3, because it appears in every third position, glycine is appearing every third position. So 1/3 of this is glycine, and that means about 33.3% of amino acids is glycine. So in 1000 it will be around 333 glycine molecules, and in X position there are 100 of them are proline, and in the Y position there are 100 of them are hydroxyproline. So together, so we have around 533 amino acids contributed only by glycine, proline and hydroxyproline. And also in the Y position we will find lysine, and some of these lysines are hydroxylated into hydroxylysine. We are going to see how the proline and lysines are hydroxylated later. This is some information about the composition of amino acids in an alpha chain that you must be aware of, because this type of information can be tested. Alright, now based on the amino acid sequence, so we have glycine X, Y position, we have either hydroxyproline, X position we have proline. So all this presence of proline and hydroxyproline, it creates bends in the alpha chain, and also just besides this proline and hydroxyproline, so there is a glycine there, and glycine is most suitable to be accommodated in small little space that is available in the bends. So that will create a alpha helical structure to an alpha chain here in a collagen molecule. So each alpha chain has got this helical structure, although it is not a classic alpha helix. And now a triple helix, which is made because of 3 alpha helical chains which are there in a left-handed helical structure, and they will intertwine with one another in a right-handed fashion, so to form a triple helix. Alright, so the diameter of this triple helix is 1.4 nanometer. Now look at a triple helix molecule. The length of that, which is given here, is 300 nanometer, and within that the quarter staggered appearance that you are seeing here. So there is a overlap of these alpha chains with one another for about a quarter of their length, and that is what is referred as a quarter staggered alignment in the alpha chains there. And also there are gaps between these alpha chains, and that will give rise to a banded appearance in a connective tissue. So this is something that you need to be aware of here. So as I was explaining in the previous slide, so the amino acid sequence in a collagen molecule, it follows glycine XY n number of time sequence. So X and Y in this glycine XY can be any other amino acids. In that, proline and hydroxyproline can be around 100 amino acids each. And proline and hydroxyprolines, they introduce bends in an alpha chain, and glycine besides this proline and hydroxyproline can be well accommodated because of its smallest side chain, which is just H. Hydroxylation of proline and hydroxylation of lysine creates additional hydroxyl containing amino acids in an alpha chain, and they are hydroxyproline and hydroxylysine. So increase in the hydroxyl group containing amino acids in a collagen molecule, it facilitates increased hydrogen bond formation between the alpha chain, and the hydrogen bonds are the ones that will stabilize the triple helical structure or triple helix structure. Remember that. That's an important point that you need to be aware of. And collagen fibers are further stabilized by the formation of covalent cross links later, after the hydrogen bonds are stabilized in the triple helix structure. So each alpha chains in a collagen in a triple helix, they can be further covalently cross-linked, thereby the tensile strength of the collagen molecule can be increased with the covalent cross-linking process. So in a typical collagen molecule, as I have already explained you, there is 33% is glycine, about 10% is proline, which is further hydroxylated into hydroxyproline. Proline and hydroxyproline, they introduce sharp bends in the alpha chain, and collagen are very resistant to digestion by proteases because the peptide bonds which are linking one amino acid with the other amino acids, because of this alpha helical formation and the twisting of alpha helical chain in a right-handed triple helix, which will make the peptide bonds inaccessible for proteases to break them. That's why collagen is very resistant to digestion by proteases. And one other important point that you must be aware is these collagen fibrils are arranged in a quarter staggered alignment, where at least a quarter of length of the collagen fibril will overlap with the other collagen fibril in that longitudinal arrangement there. And also there will be a banded appearance in these collagen fibers because of regularly spaced gaps between the collagen fibers. Now, moving on to biosynthesis of collagen. Fibroblasts are the ones that will synthesize collagen and secrete collagen into the extracellular matrix. And collagen is synthesized by ribosomes, which are present over rough endoplasmic reticulum. Once alpha chain is synthesized by ribosomes present over rough endoplasmic reticulum, this alpha chain, it will passes into the lumina of endoplasmic reticulum and then into the Golgi apparatus before it is being secreted. So, as it is going from the ribosomes into the lumina of endoplasmic reticulum and going into the Golgi complex and then entering into the extracellular matrix, and in the extracellular matrix, there are so many processes that will go on which are referred as post-translational modification. Some of the post-translational modification that will happen over collagen molecule, to name few, there is hydroxylation, glycosylation, proteolytic cleavage and cross-linking process. And many of these modifications are very unique to collagen molecule and not seen in other proteins in the body. So this particular slide is giving you overview of how a collagen biosynthesis will go on, and it also gives you the subcellular locations for each steps of collagen biosynthesis. Now the polypeptide chain synthesis is going on the ribosomes present over rough endoplasmic reticulum. So, and that polypeptide chain is entering into the lumina of rough endoplasmic reticulum for post-translational modification, and pro-collagen triple helix formation is going on as the alpha chains are interacting with one another, 3 alpha chains coming together making a pro-collagen triple helical structure, which happens. So, as the alpha chain moves from rough endoplasmic reticulum into the Golgi complex, so, and then from the Golgi complex, the secretory vesicles will go and fuse with the membrane and it will be exocytosed. So, the secretion happens, and in the extracellular matrix, there will be removal of extension peptides to make it as pro-collagen molecule to be converted into tropocollagen, and this tropocollagen will undergo further post-translational process called collagen cross-linking, and there forms a mature high tensile strength collagen fiber. So this is how rough endoplasmic reticulum, Golgi complex in the fibroblast and later in the extracellular matrix. So many processes will be going on over alpha chains, followed by triple helix, followed by pro-collagen, followed by tropocollagen, followed by collagen fiber formation happens. Now let's look at steps in the collagen biosynthesis. First thing is polypeptide synthesis. Fibroblasts are the predominant cells that will synthesize a collagen molecule. Ribosomes present over rough endoplasmic reticulum, that's where a pre-pro-alpha chain is synthesized. Now note that this pre-pro-alpha chain has got amino terminus which contains a signal sequence which contains around 15 to 30 hydrophobic amino acids, and this signal sequence will direct the pre-pro-alpha chain to enter into the cisterna of endoplasmic reticulum, where it is allowing it to go into lumina of endoplasmic reticulum. So, in the lumina of endoplasmic reticulum, pre-pro-alpha chain is converted into pro-alpha chain after removal of signal sequence. Now, the step 2 is post-translational modification. So, post-translation modification that happens first is the hydroxylation process, where pre-pro-alpha chain is hydroxylated at randomly selected proline and lysine residues. And this proline and lysine residues in the endoplasmic reticulum, so they will undergo hydroxylation catalyzed by enzyme prolyl hydroxylase and lysyl hydroxylase enzymes to introduce hydroxyl group, as it is shown here in the reaction. So, the proline residue, which is part of a polypeptide chain, that is pre-pro-alpha chain, so randomly selected, there is no selectivity there. So, ascorbate acts as a coenzyme for prolyl hydroxylase, remember that. So, a molecular oxygen is entering into this reaction and the hydroxyl group is introduced. An atom of oxygen is introduced to make it as a hydroxyl group and makes 4-hydroxyproline residue in a polypeptide chain. This is how a proline is converted into hydroxyproline, while alpha-ketoglutarate is converted to succinate. The reaction is catalyzed by prolyl hydroxylase and it is located in the lumina of rough endoplasmic reticulum. Similarly, lysyl hydroxylase does the same thing for lysine to convert that into hydroxylysine. So, this is how a hydroxylation process goes on, and importantly remember, ascorbate is a coenzyme which acts as an antioxidant there and keeping the ferrous iron in its reduced state. Now, the other type of post-translational modification that is going on in the endoplasmic reticulum is the glycosylation. See, the glycosylation over 5-hydroxyproline happens in the endoplasmic reticulum, and the glycosylation uses galactose or a disaccharide glucose galactose to be added over 5-hydroxylysine molecule, and this requires manganese and UDP glycosyl transperases. Glycosylation happens on individual pro-alpha chain and it is inhibited by triple helix formation. That means glycosylation happens only in the endoplasmic reticulum, and once triple helix formation is initiated, glycosylation is inhibited. And the precise significance of glycosylation is not yet known. Come to the third step in the collagen biosynthesis is procollagen triple helix formation. So, in the endoplasmic reticulum, look at the subcellular location. So, in the endoplasmic reticulum, 3 pro-alpha chains, they assemble together to form a triple helix procollagen molecule. So, what will happen is these pro-alpha chains, they will have N and C terminal containing extension peptides which are rich in cysteines. So, how this triple helix formation is initiated is, so at the carboxy terminus, see the cysteine amino acid having the thiol group in one of the pro-alpha chain interacting with another cysteine amino acid thiol group, so and make a disulfide linkage by oxidation, and that makes 2 alpha chains coming together, and then the third alpha chain also makes a similar linkage, disulfide linkage, bringing 3 chains together, and the interchain disulfide linkage will continue starting from at the C terminate, which is initiating the triple helix formation and goes all the way towards the N terminus, and this is how procollagen formation is initiated in the endoplasmic reticulum. And the procollagen with extension peptides is packaged into secretory vesicles within the Golgi apparatus, and then they are secreted out of fibroblasts. Now, the fourth step in collagen biosynthesis is procollagen conversion into tropocollagen formation by removal of extension peptides. So, as this procollagen molecule enters into the extracellular matrix, so there are enzymes called procollagen aminoproteinases and procollagen carboxyproteinases, as the name says. So they are going to remove C-terminus propeptide extension and N-terminus propeptide extension and convert procollagen into tropocollagen. So removal of that extension peptides at the amino terminus done by amino terminus procollagen aminoproteinases, and C-terminus extensions are removed by procollagen carboxyproteinases. So with this, procollagen is converted into tropocollagen. Now the tropocollagen molecule contains about 1000 amino acids per chain. Now the fifth step in collagen biosynthesis is cross-linking and collagen fibril formation. So now procollagen is converted into tropocollagen in the extracellular matrix. So this tropocollagen will undergo extensive cross-linking process to make a mature collagen fibrils. A staggered array of thousands of tropocollagen molecules are found in a collagen fibril, and they will have a tensile strength of collagen fibers that is provided by covalent cross links which are made between and within tropocollagen molecules. So the collagen cross linking, it will be done by an enzyme called lysyl oxidase. Remember the name here, lysyl oxidase. Don't get confused with lysyl oxidase with other enzyme which is involved in hydroxylation of lysine called hydroxylysine. Okay, so lysyl oxidase is the one which needs copper and pyridoxal phosphate, whereas lysyl hydroxylase it needs ascorbate and it is involved in hydroxylation process. So lysyl oxidase, copper and pyridoxal phosphate, this is involved in collagen cross-linking process. Let's look at what exactly happens in collagen cross-linking. So it is shown here that collagen cross-linking form through the action of copper PLP dependent lysyl oxidase. As you see, the lysine residue, it is converted into a lysine residue, where an atom of oxygen is introduced and ammonium ion is removed. Basically the amide nitrogen there present in the side chain of lysine, it is removed and released as ammonium ion, and aldehyde group is introduced in that position. That's why it is referred as alysine residue. So the lysine is converted into alysine done by lysyl oxidase. Within the polypeptide chain this happens. So what happens with the alysine there? So there can be two types of collagen cross-linking formation. One is shift base formation, where in one of the alpha chain, where the lysine residue is undergoing oxidation by lysyl oxidase, making alysine, which is having that aldehyde group, and the other alpha chain in that tropocollagen molecule with unmodified lysine. So unmodified lysine having amino group in the side chain, and that amino group is going to interact with the aldehyde group and making a shift base formation, and the shift base formation is a strong covalent linkage, and that means two chains are tied together with a strong bond and the shift base formation. That's one type of collagen cross linking. The second one next is aldol condensation. In the aldol condensation, what happens is one of the modified lysine, that is alysine, in one of the alpha chain of tropocollagen molecule, and another alpha chain of tropocollagen molecules, another alysine. So two alysines coming together, making aldol condensation there, and this is again a strong link between two alpha chains in the tropocollagen molecule. So like this, two covalent linkages, that is shift base formation and aldol condensation, they will make a collagen highly tensile strength molecule, and that's how a tropocollagen is matured into collagen fibril. Alright, now you can see propeptide extensions which are taken out, converting procollagen into tropocollagen molecule, and tropocollagen is undergoing a cross-linking process and making a mature collagen molecule with the help of lysyl oxidase enzyme. So this slide is showing you steps in collagen biosynthesis, especially with special attention to location with each processes that are going on in the collagen biosynthesis. Make sure you know each and every processes that are going on in each subcellular organelles here. Very important. Rough endoplasmic reticulum, there is synthesis of pre-procollagen, insertion of the procollagen molecule into the lumen of endoplasmic reticulum. That's what happens. And in the lumen of endoplasmic reticulum, there will be hydroxylation of proline and lysine residues, and then glycosylation of selected hydroxylysine residues happens. And in the lumen of endoplasmic reticulum, going into the Golgi complex, there will be self-assembly of tropocollagen molecules. So in the lumen of endoplasmic reticulum and Golgi apparatus, so there will be self-assembly of procollagen molecules initiated by disulfide bond formation starting from the carboxy terminus extensions going all the way towards the amino terminus, and there forms the triple helix molecule. So procollagen has the triple helix now. So secretory vesicles will have that procollagen which is prepared for secretion from the cell into the extracellular matrix. Now in the extracellular matrix, what happens is, so the cleavage of the propeptides and then removing amino and carboxy terminal extensions happen, and then the self-assembly of the collagen molecules into fibrils, and then the mature collagen fibers happens with the help of lysyl oxidase. So these are all the subcellular locations and the processes that goes on. Make sure you remember this. Now, dividing into intracellular processes and the extracellular processes in the order and location of processing of the fibrillar collagen precursor. So intracellularly what happens? Cleavage of the signal peptide coming from the endoplasmic reticulum as a pre-pro alpha chain, and then hydroxylation of prolyl residues, lysyl residues, and then the glycosylation of hydroxylysyl residues happens inside that, and then the formation of intra-chain and inter-chain disulfide linkages happens, and the formation of triple helix procollagen happens inside the cell, intracellular. Now the extracellular is cleavage of amino and carboxy terminal peptides, assembly of collagen fibers in a quarter staggered alignment. Okay, so the oxidative deamination of side chain amino group of lysyl and hydroxylysyl residues to their aldehyde groups like allysin residues happens outside, and then formation of intra and interchain cross links like Schiff base and aldol condensation happens to make a mature collagen fibril. So this is how order and location of processing of the fibrillar collagen precursor into mature collagen is done. Now let's move on to see briefly a degradation of collagen molecule. Collagen tissue undergoes constant but slow turnover. The degradation occurs during starvation, tissue repair, and during normal growth and development, and this is initiated via enzyme initiated collagenases. Collagenases are the enzymes that will degrade a collagen molecule, although it is a slow process. Collagenases are extracellular zinc containing proteinases which will split a bond between glycine and isoleucine. And the tumor cells secrete large amount of collagenases that help them in metastatic spread. Remember that clinical application there. So as in the flow of reaction, collagen by collagenases break that bond between glycine and isoleucine and release a polypeptide fragments, and then cathepsins will break that polypeptide fragments into peptides, and the peptides are further handled by proteases. So this is how degradation of collagen will go on. Coming to classification of collagens according to the structure they form. So collagens can be classified into fibril forming collagen, and the type of collagens are mentioned in the numbers there. Then the network forming collagens, facets, multiplexins, beaded filaments, anchoring fibrils, and transmembrane type of collagen. One of the type of collagen that I would like to make a special mention is network forming collagen, which are type 4, type 8 and type 10 collagens are network forming collagen, and importantly type 4 collagen is found in the glomerular basement membrane, and that helps in the glomerular filtration with the help of heparan sulfate. Now, some collagens, they do not form fibrils, so they are characterized by interruptions of triple helix with stretches of proteins lacking glycine XY repeat sequence. These non-glycine XY sequences result in areas of globular structure instead of alpha helical structure, which are interspersed in the triple helical structure. So, in that we have network-like collagens like type 4 collagen, which form the basement membranes, and then fibril-associated collagens with interrupted triple helix, that is referred as facets. They have interruptions in the triple helical domain because of non-glycine XY sequence appearance. Beaded filaments, they consist of long chains of collagen molecules which have got regular beaded appearance. And then we have anchoring fibrils like collagen type 7 forms the main part of anchoring fibrils in the epithelial tissues. Transmembrane collagens, they have a short intracellular N-terminal domains and extracellular domains with long interrupted triple helixes. And multiplexins, these are the collagens with multiple triple helix domains and interruptions. So this is just an example of where these different types of collagens are found. So this is about connective tissue. In that we have covered extensively on the collagen molecules, including different types of collagens, collagen biosynthesis, post-translational modification, and all the details, including subcellular location, processes, enzymes involved, and all the details. Thank you.